Chlamydia is present in the form of elementary body when it is outside the host cell, then transformed into a reticulate body upon inclusion into the host cell. Now we shall look at the life cycle of this particular organism and understand how it is able to propagate among human beings. Firstly, when an elementary body gradually approaches a host cell, its MOMP situated on the surface would interact with a specific receptor located on the host cell membrane. Here we talk about the specificity of Chlamydia Trachomatis because it only targets the columnar epithelial cells. In this case, the MOMP would specifically recognize sialic acid receptors on epithelial cell membrane. Once the cell-cell reconnection is completed, the elementary body will then form a vacuole within the host cell and intrude the cell via endocytosis. At this point, our cell defense mechanism, which is the lysosome, will be activated to kill the intruding elementary body. However, a set of type 3 secretion apparatus, which composes of needle-like projections present on the elementary body itself, would secrete protein fragments into the host cytoplasm and block the action of lysosome. This is one of the ways for the elementary body to stay alive in the host cell. Besides, it can also take in diverted lipids from the host Golgi apparatus and form a thin lipid layer around the vacuole. This mechanism is able to make the elementary body to become indistinguishable with other cell organelles in the host cell, hence escape from being hydrolyzed by enzymes present in the lysosome. Okay, now, once the elementary body is secured from outer metastasis, now it will modify itself to become a bigger, non-infectious but reproducible reticulate body. By using ATP from the host cell, the reticulate body is able to undergo binary fission and replicate up to 1,000 cells. After the replication, the reticulate bodies will then transform back to elementary bodies to be transported out of the host cell. We are actually going to look into two different exclusion mechanisms of the elementary bodies. First way is where most of the reticulate bodies are transformed back to elementary bodies and leave the cell by exocytosis to infect other cells. The remaining reticulate bodies will stay and continue the cycle. The next mechanism involves the role of bacterial proteases. Every reticulate body is modified back to elementary body, and with the help of the proteases, the host cell would lyse apart, releasing all the elementary bodies into the extracellular environment to spread the infection.